All right. Questions over what we, so let's just talk about the food journal thing. So either turn in your food journals here at the office or add them, submit them in the assignment that is on the Moodle site. I was getting my other class to submit things to me via email, and then I discovered that very quickly filled up my email box in terms of the size of the files, and so I'm making all assignments on Moodle so that I keep my inbox a little freer. So no food journaling over the weekend, and today I will post a new set of food blogging prompts for you for the next 2 weeks. And just to remind you, the rules, unless there's something different stated on the Moodle site, are 2 posts per week and 2 comments on other people's posts per week. And yeah, that's that. Questions about food journaling, food blogging? All right, cool. Questions about what we were covering last time. We finished up with mitosis. I felt like it was a very lecture-y kind of day. Do you have any questions about that material? No. Okay. So how many cell divisions does mitosis go through? Blake. I can pick on Blake because I can't see his face. Still can't see his face. Blake.
Sorry, I was trying to write food journal.
Oh, okay.
What'd you ask?
How many cell divisions in mitosis?
Let's see. Is there 4?
There's not 4. How many?
Are you talking about the phases? How many phases there is? Like the.
So does it go through 1 phase of cell division, or does it go through 2 phases of cell division?
Goes through 1, right?
Yeah, it goes through 1. It divides from 1 cell into 2 cells.
Yeah.
Branson, are the cells at the end of this first cell, this 1 cell division in mitosis, are those going to have the full complement of diploid chromosomes, or do those chromosomes get cut in half in terms of number? Can't hear you. Press your space bar.
Will they cut in half?
What's that? They're not cut in half. Nope. You start out with 46 if you're a human, and you end up with 46 at the end. So you go from diploid to diploid. McKinsey. What are the major things to look for in prophase? If you were looking at a cell in prophase, what would you see?
Prophase?
Yeah, in prophase.
It would just, the first phase?
That's the first phase.
They'd just beginning to connect, right?
Well, the chromosomes are getting, what's their size doing?
Their sides?
The size of the chromosomes?
Oh, size. They're beginning to go from 23 to 46.
Nope, they've already done that. They've done that back in the S phase of the cell cycle. What's happening to them in terms of their physical size of the individual chromosomes?
Are they getting ready to split?
They're getting ready to split, but during the S phase, they're really long and strung out. What happens to them during prophase?
They shrink, and then they get thicker or longer and thicker.
They get shorter and thicker. Yeah. Okay. What else happens in prophase? Somebody else. Marcus.
Is new nucleotides added to the exposed nitrogenous?
Nope. Once again, that happens during DNA replication during the S phase of the cell cycle. So in prophase, the chromosomes get shorter and thicker, and then some other things happen that connect to those chromosomes. What things connect to the chromosomes during prophase?
New strand of DNA uses old.
What's that?
New strand of DNA uses old.
Nope.
No.
Somebody help Marcus out.
Isn't it spindles?
Yeah, those spindle fibers. Those spindle fibers connect to the centromeres of the chromosomes. Winston, what would we be looking for if we saw? What would we be seeing if we saw a cell that was in metaphase, which is the next phase of mitosis? I can't hear you. Are you muted?
Yeah, my bad. I was muted.
That's all right.
Is that when those chromatids, they break in half and then they go to each side of the cell?
So that's in anaphase. That's the next
Oh.
phase. But before you get there, somebody tell me what you would see in metaphase.
Don't you see like a plate, like a metaphase plate?
Yeah, who's that? Olivia. Okay, yeah. Yeah, so you would see the chromosomes all lined up along the middle of the cell in a line, and they do that because they're connected by the spindle fibers, and the spindle fibers arrange the chromosomes in the middle. And then after that, they break apart and get pulled apart by those spindle fibers, which is what Winston was talking about. And so that's in the next phase, which is anaphase. So who can describe for me what's happening in telophase?
It's the formation of the new Celsius, is that right?
The new nucleus, yeah. So the new nuclei with the chromosomes inside of them, and then also the cytoplasms begin to separate as well to form the two new cells and the two new daughter cells. So that is kind of a brief review of mitosis. And the reason that we normally teach mitosis before we teach meiosis is because meiosis is more complicated, but the second half of meiosis is just like mitosis. So they both share some similarities, but they're also fundamentally different in some ways. Let me pull up the PowerPoint and then we can take a look at it. All right. Can you guys see my screen with all of the slides on it?
Yeah.
Okay, cool. Whoa. All right. So this is what we were talking about. During prophase, the chromosomes get shorter and thicker, and also the nuclear membrane that hems them in begins to break down. During late prophase, that has completed. There is no longer any nucleus. During metaphase, you see that the chromosomes are lined up along the metaphase plate. They're connected to the centrioles by spindle fibers. In anaphase, the two chromatids separate and start migrating towards where the new nuclei are going to be. That's what anaphase is about. And then in telophase, the new nuclei are forming. And once again, remember that this figure has the wrong number of chromatids in telophase here. So I was putting a chart on the board for you where we have a contrast of mitosis versus meiosis. And we can think about this in terms of the function. What does mitosis do? And mitosis is used for what we refer to as somatic growth. This is the growth of the body, so it's producing new cells that are identical cells. And I told you last time that meiosis is the way we produce gametes. We can break this down into the number of cell divisions. there is one cell division in mitosis and there are two in meiosis. We can break this down into the number of daughter cells. How many cells are produced at the end of mitosis? Anyone? We already answered this earlier.
2.
Yeah, we have 2 daughter cells, and these daughter cells are diploid. Meaning that they have 2 copies of each chromosome. And if we think about the genetic diversity of the daughter cells, in mitosis, all of the daughter cells are genetically identical. So what we'll do today is, as we go through meiosis, we'll see how meiosis differs from mitosis. So everybody with me? So in meiosis, you have to go through the steps of preparing the cell the same way you would from mitosis. You have to duplicate all the chromosomes. And in prophase, once again, these chromosomes are going to go from these long strung out chromosomes to shorter and thicker chromosomes. But one fundamental difference is that chromosomes that are alike, that are what we refer to as homologous chromosomes, a homologous chromosome is 2 chromosomes that are of the same type. So if we go back and look at the karyotype, you have 2 copies of chromosome 1. You have 1 copy that came from your mom, another copy that came from your dad. Those 2 copies of chromosome 1, those are homologs, 2 different versions of chromosome 1. You have 2 different versions of chromosome 2, 2 versions of chromosome 3. So you have 23 homologous pairs in a diploid organism if you're a human. If you're a different type of organism, like for example, if you're a fruit fly, you have 4 pairs of homologous chromosomes. So this represents the homologous chromosomes, and so the genes on this homolog are the same as the genes on this homolog, even though you might have different forms of that gene. So I'm going to get all of you on screen for a minute. So I can see all of you. Do you guys know how to roll your tongue? How many of you can roll your tongue? Let's see it. Okay, some of you can't. Who can't roll their tongue? All of you can roll your tongue?
I can't.
Who's that?
Blake.
Blake. So Blake cannot. Who else?
I can't roll mine.
Is that Marcus?
Kenton.
Oh, Kenton. Okay. All right. So tongue rolling is a genetic trait. Some people can roll their tongue and other people cannot roll their tongue. So there's variation in the trait, and it's controlled by a single gene. So there's a single gene that enables you to do this or not. Luckily, because I teach genetics sometimes, I have the ability to roll my tongue, and so I can illustrate this for people who can't. And so if we go back to that figure, if we go back to this figure, let's say that the tongue rolling gene occurred right here. Well, there are 2 different varieties of the tongue rolling gene. One is the dominant variety, which allows you to roll your tongue, and the other one is the recessive form of that gene, which prevents you from rolling your tongue. And so you get these traits from your parents. So if you can roll your tongue, chances are one of your parents can also roll their tongue. And so what you have here is you might have one parent who can roll their tongue, and they would have one form of the gene on one of the homologs. But if your other parent couldn't roll their tongue, they have two copies of the allele, the form of the gene, that prevents them from rolling your tongue. And so you would have one copy of a tongue rolling gene and one copy of the non-tongue rolling version of that gene. And they exist on the two homologs. So the homologs can have different forms of the genes, but the gene for tongue rolling is on this one chromosome. Does that make sense? So what happens in meiosis is these homologous pairs pair up with one another. And this process of pairing up is referred to as synapsis. Synapsis is the process of these chromosomes pairing up with one another. Now, that's the fundamental difference between prophase 1 of meiosis and prophase of mitosis. The chromosomes shrink, but as they shrink, they go through this process of synapsis. One of the other things that you'll see in this drawing that's very accurate is that during synapsis, the different arms of the chromatids cross over with one another. And this becomes very important from genetic diversity. So this process of process of crossing over occurs during synapsis. And what can happen during this crossing over is that you get interchanges of the chromosomes between the two chromosomes. So when this green chromosome crosses over with the blue chromosome, what can happen is the green part can break off and become attached to the mainly blue chromosome, and the blue part can come apart and attach to the mainly green chromosome. So now, if you had there's my markers. So can you guys see my board okay? Say yes, because I can't really see you from here.
Yeah.
Okay, so if you have an orange chromosome, the two chromatids of the chromosome are connected to one another, and you have a blue chromosome. Let's say the blue chromosome came from your mom and the orange chromosome came from your father. And you're going through the process of meiosis, and on this chromosome, let's say that this is chromosome 21 in the human genome, and on this chromosome, there is a gene for the tongue rolling gene, and it occurs right here. And your father has the ability to tongue roll, so we'll give him a capital T, but your mother does not have the ability to tongue roll, and so she gave you the recessive version of this gene. When crossing over occurs and you get the interchange of genetic material such that the blue part of your mom's chromosome becomes attached to your father's chromosome over here, and the orange part of your father's chromatid becomes a part of your mother's chromosome, it carries with it the genes. So now this chromosome with its two chromatids has both versions of the tongue rolling gene, and this chromosome with the two chromatids has both versions of the tongue rolling gene on it. So this genetic interchange, this interchange of chromosomes, leads to this allele here. These different varieties of a gene are called alleles. Different forms of a gene are called alleles. This allele now becomes associated with a bunch of genes that it wasn't associated with before. this allele now becomes associated with a bunch of genes that it wasn't associated with before. And so this process of synapsis and crossing over mixes up which genes co-occur with one another on a chromosome. And so the reason that I've used this particular figure is because it shows this mixture of things throughout the whole panel. And so we can track whether or not at the end, whether the daughter cells are actually genetically identical or not. So what's the diploid number of chromosomes in this particular example? How many chromosomes do we have total? Anybody? I can't see you. I can't hear you.
8.
We don't have 8. What do we have instead? Who was that?
Is it 4?
We have 1 chromosome, 2 chromosomes, 3 chromosomes, 4 chromosomes. Now we have 8 different copies of DNA because each of these chromosomes is made up of 2 different chromatids. But remember, we have this weird terminology around chromosomes where 2 chromatids that are bound together by a centromere are basically 1 chromosome. So we have 1, 2, 3, 4 chromosomes. We have 8 copies of DNA because we have 8 chromatids. So the diploid number of chromosomes is what in this example?
Four.
Yeah, the diploid number is 4 because we have 2 copies of 2 different kinds of chromosome. We could call them chromosome 1 and chromosome 2. Or in this case, we could just call them the short chromosome there and the long chromosome there. So what happens then during meiosis is these chromosomes go through synapsis. They interchange the ends of their chromosomes with one another. And then in metaphase 1, so remember we have prophase 1, we put a 1 and a 2 at the end of these phases to distinguish whether we're talking about the first phase of meiosis or the second phase of meiosis. So in metaphase 1, these chromosomes that are paired up, that have gone through synapsis, they line up along a metaphase plate, just like you would have in mitosis, except that in mitosis, when they line up along the metaphase plate, they're not paired in homologous pairs, but in meiosis, they line up along the metaphase plate in a paired fashion, where the long chromosomes are paired together and the short chromosomes are paired together. Then in anaphase 1, it drags one set of chromosomes to one new cell and another set of chromosomes to the other cell, but the chromatids don't separate from one another. In this case, the homologous pairs separate from one another. And then in telophase 1 or prophase 2, you now have 2 new cells. At this point, each of those cells has how many chromosomes in them?
Or 2.
They have 2. This cell has 2, this cell has 2. They have 1 copy of the short chromosome and 1 copy of the long chromosome. And so these cells have now gone from being diploid, having 2 copies of each type of chromosome, to becoming haploid, meaning that they have 1 copy of each type of chromosome. This is the difference between haploid and diploid when we talk about ploidy. So what they've done is they've just taken the 1 copy of each homologous pair, partition 1 copy of each homologous pair into one cell and the other copy into the other cell. During metaphase, you see that they line up along metaphase 2, they line up along a metaphase plate. Both cells are going through this now at the same time. They line up along the metaphase plate, and in anaphase 2, Now the two sister chromatids get separated into the different cells, and then you end up with 4 cells. Each cell has how many chromosomes?
Who?
Each cell has 2 chromosomes, and the chromosomes just have a single chromatid each one, because that's the way it's supposed to be. So meiosis goes through 2 phases of cell division, meiosis 1 and meiosis 2. It starts off with diploid cells, ends up with haploid cells, and rather than having 2 daughter cells at the end, it has 4 daughter cells at the end. And the other thing is, when you look at this, each of these chromosomes is slightly different from all of the chromosomes in the other cells, because they've gone through this interchange of chromosome pieces back here during synapsis and crossing over. So there is a mistake in this figure also. This is once again a textbook figure. Where is the mistake in this figure, in the way that these chromosomes are drawn?
In metaphase 2, there.
Yeah. What's wrong with metaphase 2 drawing?
No sort of variation.
Oh, repeat that.
There's no sort of variation. It doesn't. It hasn't gone through the interchange.
Yeah. Okay.
It hasn't
So
gone through the genetic interchange in metaphase
Yeah.
2, but it like has in the telophase prophase picture.
Yeah.
Just like all of a sudden.
Yeah, so this figure that is prophase 2 and this figure that is anaphase 2 are correct, and the artist lost track of what he or she was doing and didn't do the genetic interchanges the correct way. And so you once again make a note of that. These should have the same crossover pieces that the other figures do as well. But what that crossing over does is it basically now has this blue chromosome associated with a bunch of the gene varieties that is on the green chromosome. This little section of green chromosome is associated with blue chromosome varieties of the genes. And so this process of going through meiosis, not only does it produce more daughter cells, but those daughter cells are genetically different from one another. So this creates genetic diversity, which is one of the things that it would be nice to have when you're creating offspring. You don't necessarily want all of your offspring to be exactly alike. And so when we think about the number of daughter cells, in mitosis, we end up with 2 daughter cells. In meiosis, we end up with 4. The cells that we end up with in mitosis are diploid. These are haploid. And instead of them all being identical, and I have spelled identical incorrectly, these are all genetically unique in terms of the forms of the genes that they have on them. So this is roughly how meiosis works. This is a table that shows you kind of what some of the major features are that separate these two. But I also want you to be able to, on a test, be able to recognize if I give you a cell that is in metaphase 1 of meiosis, how would you know that it's in metaphase 1 of meiosis versus metaphase of mitosis?
Whether or not they've got the homologous pairs.
Yeah, so meiosis will show homologous pairs, whereas in mitosis, those homologous pairs are not paired up like that. They still are along a metaphase plate, but they're not paired as homologous pairs. If we were looking, if I showed you a photo of metaphase 2, how would you separate that from metaphase 1 or metaphase of mitosis?
You can separate it from metaphase 1 because, again, there's no homologous pairs.
Okay, so it's not metaphase 1 because there's no homologous pairs. How does it then differ from metaphase of mitosis? Somebody other than Kenton.
Because of the number of chromosomes in each cell.
Okay, so the number of chromosomes, so I would always have to tell you what the diploid number of chromosomes is of the organism doing it in order for you to figure this out. What else would clue you in that you're not in mitosis? How many cells are undergoing cell division in metaphase 2?
2.
There are 2 cells undergoing cell division in metaphase 2. So if you knew that the diploid number of chromosomes was 4, you would notice that there are 2 cells undergoing cell division, and each of those 2 cells only had 2 chromosomes. All of that would clue you into the fact that you were in the second phase of meiosis rather than in some phase of mitosis. So my advice in studying this material is to study it as a process, study it in order. But then while you're studying it in order, since you now have been exposed to both mitosis and meiosis, ask yourself, okay, I'm in meiosis. How does meiosis at this phase differ from similar phases in mitosis so that you can keep those 2 processes straight? Questions about that? Okay, so this is how gametes get formed. So your body goes through the process of creating gametes. If you're a male, it happens in the testes. If you're a female, it happens in the ovaries. And you begin with regular somatic cells, regular body cells. But in the process of going through meiosis, you basically create a bunch of cells that have half the number of chromosomes. And then when fertilization occurs, when you have sex with somebody and create a baby, you basically combine your half of your chromosomes with the half of chromosomes that come with the person that you mated with, and then that creates a full complement of chromosomes that are found in your offspring. So you are a diploid organism that produces haploid gametes that then go to produce a diploid organism in your offspring. And that's how your offspring get the gene varieties that you possess in them. And so half of the genes, half of your genetic complement you got from your mother, and half of your genetic complement you got from your father. And it's the same with corn, it's the same with soybeans, it's the same with everybody. So now, in order to kind of understand genetics and how we go from genes to what a corn plant or a soybean plant or a human being look like, we need to understand some things about what happens to these genes, what these genes are used for. So there's a process that cells go through which basically takes the genetic information in genes and converts that genetic information into what we call the phenotype. So the genotype refers to the combination of different forms of the gene that you have, and that gets translated into the phenotype. And the phenotype is kind of the outward expression of those genes. So remember that for any given gene, you have 2 versions, two copies of that gene. One copy you got from your mom, one copy you got from your dad. If those 2 copies of the gene are the same, so for tongue rolling, you either have 2 versions of the gene that code for having the ability to tongue roll, or you have 2 copies of the gene that prevent you from tongue rolling, or you can have 1 copy of each. This is the genotype. It refers to what's the actual identity of the genes on the chromosomes. So for the tongue rolling gene, you either have 2 tongue rolling genes, 1 tongue rolling, 1 non-tongue rolling, or 2 non-tongue rolling versions of the gene. These are genotypes. Because you're a diploid organism, you have 2 copies of each gene. In this case, if you possess this genotype or this genotype, the phenotype that you have is that you are a tongue roller. But if you have this genotype, you are not a tongue roller. And this is the phenotype. The phenotype is the outward expression of the genotype. So the outward expression is you can either do this or you can't. And so because if you have the big T version of the tongue rolling gene, this is referred to as the dominant allele, and this is referred to as the recessive, because when it's in the presence of the dominant, it recedes in a sense. It doesn't express itself. The only time that you get recessive traits being expressed is when they occur in the condition where there's 2 of the same kind. When there's 2 of the same kind, you are what we refer to as homozygous. So this is a homozygous dominant genotype. This is a homozygous recessive genotype. And this genotype in the middle is what we refer to as heterozygous, where you have 2 different alleles of the same gene. Now I realize I've just loaded a bunch of new terminology onto you, but hopefully you can see all of that. Can you see all of that? Okay. These terms are also in your textbook in the section on genetics, so you can also go and find it there as well. So after you have become an individual, your cells begin to divide, and as they're dividing, the genes that you possess get used. They get what we call expressed. The information that is in the sequence of nitrogenous bases in DNA get converted into RNA, and then the sequences of RNA are used to make proteins. And so you need to understand a little bit about DNA and RNA and protein synthesis in order to understand some things about genetic engineering that we're going to be covering later. So that's the reason that we're kind of going through this level of detail. So when you want to make a protein, the first thing that you have to do is you have to take the information in DNA and convert it into the language of RNA. So what we basically do is we do something similar to what we do when we make new DNA. So if you go back a number of slides, we talked about making new DNA. The first thing you do when you make new DNA is you have to split the double helix of DNA. And then what you do is you build new DNA on one side of the split, you build new DNA on the other side of the split. When you make RNA though, you basically take the DNA molecule and you split it up and you build RNA on 1/2 of the DNA molecule. And so you undergo complementary base pairing, but back during DNA synthesis, there were 4 nucleotides, 4 different nitrogenous bases. The nitrogenous bases were what? Do you guys remember?
Those were like the AT things that connected to each other, right?
Yep, adenine, thymine, guanine, and cytosine. These are the 4 nitrogenous bases, those nitrogen-containing compounds that are in the middle that connect the 2 halves of DNA. And remember that adenine always bonds to thymine and guanine always bonds to cytosine. This is what happens in DNA, but what happens in RNA is you have a different kind of sugar So DNA and RNA stand for deoxyribonucleic acid and ribonucleic acid. So deoxyribose is one type of 5-carbon sugar. Ribose is another type of 5-carbon sugar. So they differ in the type of sugar that they have, but they also differ in that RNA has adenine, guanine, and cytosine, but in RNA, thymine is replaced with a different nitrogenous base called uracil. And so in this figure, you see that this thymine is bonded to an adenine, this cytosine is bonded to a guanine, this guanine is bonded to a cytosine, but this adenine now is bonded to a uracil. So the same kind of complementary base pairing happens in the adenine complementary base pairs with uracil and guanine complementary base pairs with cytosine. Okay. And the end result of this is that you end up with a single strand of RNA, and after the RNA has been created, the DNA just goes back and heals up itself and complementary base pairs with the other half strand of DNA. And so what RNA synthesis does is it unzips a little bubble of the DNA, builds the RNA inside of that bubble, and then puts that DNA back together. And then the RNA is transported to the cytoplasm, and that's where protein synthesis occurs. But the process is actually very similar and analogous to DNA synthesis, except that you're creating new strands of DNA here, but a lot of the mechanics are very similar. So once you have an RNA molecule, the RNA molecule has the complement of the genetic code, the ordering of these nitrogenous bases that is found in the DNA. And what happens in protein synthesis is this messenger RNA molecule that you've created is used by a structure called a ribosome to basically build a protein. Now this is super complicated, and so I want to walk you through this step by step. So a messenger RNA molecule is this long strand that we just built from DNA. It has a beginning and it has an end. And at the beginning of protein synthesis, the ribosome occurs in two sections. There's a big section that is on top, and there's a little section that is below. And what happens is these two sections come together and kind of clamp down on the messenger RNA molecule. And in the messenger RNA molecule, there are what we refer to as active sites. There are places where the ribosome has enzymatic activity. And floating around in the cytoplasm are transfer RNA molecules. So we have we have messenger RNA. We have ribosomal RNA. So this ribosome has RNA in it also, and we have what we refer to as transfer RNA. So we have a bunch of different kinds of RNA. The messenger RNA is the long strand, ribosomal RNA is in the ribosomes, and transfer RNA is just floating around in the cytoplasm. And it shows you a pretty good view of what it looks like here. When I draw it, I usually draw it as a cross with a little circle at the bottom of it. And the circle at the bottom of it is a place, because it's RNA, it has RNA nitrogenous bases hanging off of it. And these RNA nitrogenous bases complementary base pair to sections of the messenger RNA that are inside this ribosome. So that what happens is that the transfer RNA can come and sit in this active site, right like this, through complementary base pairing, because as you can see in the drawing on your PowerPoint, you can see that this is a uracil, an adenine, and a cytosine, and it complementary base pairs with an adenine, a uracil, and a guanine. And so this transfer RNA molecule, what it's doing is it's transferring an amino acid to the ribosome, and it does that through this complementary base pairing. So if you have in your messenger RNA molecule an adenine, a uracil, and a guanine, the only transfer RNA molecule that will occur and match up with this is a uracil adenine cytosine transfer RNA molecule, and the only amino acid that will be attached to that is an amino acid called methionine. So amino acids are the building blocks of proteins. A protein is just a bunch of amino acids strung together. And so this transfer RNA molecule is responsible for bringing the amino acid to the ribosome so that it can be attached to another amino acid. So the example in your book, or in your figure here, shows that the second three nitrogenous base sequence is cytosine, uracil, and guanine, which means that it will connect to a transfer RNA molecule that has guanine, adenine, and cytosine. And that is a transfer RNA molecule that will possess the amino acid leucine. And so what happens is CUG, the next three sequence code here matches up with guanine, adenine, and cytosine, and that is a transfer RNA molecule that carries the amino acid leucine. Now, once those two amino acids are next to one another in the ribosome has the power to create a bond between methionine and leucine. When it does that, it breaks the bond of methionine with the transfer RNA molecule. The transfer RNA molecule gets kicked out. And once this active site here, this binding site here, is free, then the ribosome shifts down a segment, moves down the molecule. So now it's centered here on a new 3 nitrogenous base sequence. This then is a transfer RNA molecule that is bonded to leucine, and that leucine is bonded to methionine. And whatever the next 3-letter sequence is, in your drawing, it's an adenine, adenine, uracil. Adenine, adenine, uracil is now free to have a transfer RNA molecule come in here, which will have the anticodon uracil, uracil, adenine, and that then is attached to an amino acid that it doesn't say, but we could look it up. There's another amino acid here. And then what's going to happen is the ribosome is going to form a bond between leucine and this new amino acid. It's going to break the bond of this transfer RNA molecule with leucine. It's going to kick that transfer RNA molecule out, and then it's going to shift down to the next three nitrogenous base sequence. So these 3 nitrogenous base sequences on messenger RNA are called codons. The corresponding sequence on the transfer RNA is called an anticodon. And so the ribosome is centered on 2 different codons at a time. The codon matches up with the anticodon, which specifies the amino acid. And after it's bonded this amino acid to the next amino acid in the sequence, it then shifts down to encompass the next codon, which then allows the anticodon of transfer RNA to match up, which carries with it its own unique amino acid. And it just keeps doing this down a long strand of messenger RNA until you created a long strand of amino acids. And basically proteins are just long strands of amino acids bonded together with one another. It continues doing this down a long stretch of RNA until it gets to a thing called a stop codon, and the end result is that you get a long polypeptide chain, this long chain of amino acids. We know how all of this works. So for example, we saw that this next codon was an adenine uracil. This figure doesn't tell you what amino acid you can expect to come out of that, but there's this thing called a codon table where we can look at adenine uracil, and adenine uracil specifies the amino acid asparagine. And these proteins have different charges. These amino acids have different charges. They can be nonpolar or polar. Remember, we talked about polar molecules like water due to the electronegativity of oxygen. So amino acids can also be charged. You don't need to know about the codon table, except a couple of things that I want you to notice about it. There is AUG. You'll notice that in the figure in the PowerPoint, AUG is the first codon sequence here, and that's because that's the start codon. That's the codon that tells the cell that this is where protein synthesis begins. And then there are stop codons. There are 3 stop codons: uracil adenine adenine, uracil adenine guanine, and uracil guanine adenine. Not surprising that all of these things start with uracil and just differ in the last two positions of the codon. And so this is the thing that signals to the ribosome to stop adding new proteins. And the difference between the transfer RNAs that correspond to this, so if you have a uracil adenine down here in the messenger RNA, that corresponds to an adenine uracil transfer RNA molecule. What's different about this transfer RNA molecule is that there is no amino acid attached to that. And so it comes to the end of protein synthesis because the transfer RNA molecule that corresponds to that codon doesn't have an amino acid attached to it. And so it's like the lack of an amino acid on the transfer RNA molecule is actually what stops the process of protein synthesis, because when it goes to add this serine to this one, there's nothing there to add it to. So it just has nothing to connect to at that point. So that's a big, complicated process. Do you have questions about that process of going from DNA? What's that?
How will that information show up on the test?
Oh, Jesus Christ, that's a good question, because normally I might give you some figures and ask you to, for example, label the figures and things like that, but that would be on a handwritten test that I gave you in class. I'm not sure how I would ask this on an exam. I guess I would probably do it the same way that I would ask questions about mitosis and meiosis. So earlier when we first started class today, I was like, okay, can you tell me what the major events are that are happening within the cell during prophase? What are the big things that are happening in a cell during metaphase? How do you recognize when a cell is in anaphase, et cetera, et cetera? The things that I might ask you is, I might ask you, there's an RNA molecule whose role is to bring amino acids to the active sites of ribosomes. What type of RNA is that? Is that messenger RNA? Is it ribosomal RNA? Is it transfer RNA? So that would be a kind of multiple choice test that I could ask you that gets at the information contained in these things. The way to answer that question, though, is to basically learn this as a process. Once again, what's the process that we go through? the process that we go through is, and I'm going to load a little more terminology onto you at this point. Is we take DNA, we turn that DNA into messenger RNA, and then we turn that messenger RNA into an amino acid sequence. And this is how we make a protein. So how do we do this? Well, we do this by just doing a very similar process to DNA replication, only it results in a single strand of RNA. So we have double-stranded molecule being converted into a single-stranded molecule. This has adenine, thymine, cytosine, and guanine. This has adenine, uracil, cytosine, and guanine. This process here is called transcription, and I'll explain what that means here in a minute. Then this long single-stranded molecule is going to be used with ribosomes as the enzymatic engine that does the job, and transfer RNA molecules, which are how amino acids get to the ribosome. And that whole apparatus is going to be used to create the amino acid sequence. This process here is called translation. And within all of that, it would be good for you to know the mechanics of what is going on. You have these two active sites in the ribosome. The first active site is where the first transfer RNA molecule comes in. The second active site is where the second one comes in. The ribosome binds the first amino acid to the second amino acid and then kicks the transfer RNA molecule of the first amino acid out, and then the whole thing shifts down. So learn that process. In the process of learning that process, you will be using terms like codon and anticodon to describe that. You'll be using terms like transfer RNA versus messenger RNA and stuff like that. So you need to have facility with those terms as well. Once again, these codons, the information in messenger RNA, which came from information in DNA, basically specifies the order of amino acids, and the order of amino acids is important in determining what that protein is that's being built. So learn the process, and hopefully in the process of learning the process, any little question that I ask you about any part of the process, you should be able to answer. But I'll have to be careful in how I ask you questions about this process that tells me, do you have the process down well enough? And it's just I'm gonna have to be very, very thoughtful about how I phrase questions on the test in order to elicit the answers that I want from you. Does that make sense? And my goal is later on today, I'll actually put together a set of 10 or 15 questions in multiple choice format that are going to be very similar to the kinds of questions that I might be asking on the exam, to give you some feel for what the exam is going to be like, so that 2 Mondays from now, which I think is when your test is scheduled, you get to that test and you're not surprised at kind of the form of the questions. Does that make sense? That's on my list of things to do today. We call this first process transcription because we're basically, if you want to think of this as an analogy to language, here I have Michael Pollan's book. And back in the old days, if I was a monk in a monastery whose job was to transcribe books, I would open up a freshly bound book that had a bunch of blank pages in it, and I would open up Michael Pollan's book, and I would copy word for word, page by page, the text in Michael Pollan's book into a new copy of Michael Pollan's book. It's how we made books before the printing press came around. And so that process of taking a book in one language and converting it to a new book in another language is called transcription. Now, if I wanted to sell Michael Pollan's book in Japanese, I would have to give this book to somebody who spoke both English and Japanese, And they would sit down and they would open up Poland's book in English. They would open a blank book and they would take Poland's words in English and they would translate them to a new book that is written in Japanese. And so we call these two processes transcription because we're taking information in the language of nucleic acids, DNA, and we're transcribing it into a slightly altered form, but it's still the language of nucleic acids. So we're just transcribing the information in DNA into a very similar language of RNA. It would be like taking New Jersey English and translating it into South Carolina English. But translation takes the information that is pent up in nucleic acids, and it translates it into a new language, the language of amino acids and proteins. And so we call that process translation because we're moving from nitrogenous base stored information to amino acid stored information. And that's why the two processes are called what they're called. Questions? We need to talk a little bit about mutation, but we only have a couple of minutes left, so we'll cover that next time. And then we're going to need to be covering genetics. So I think if I look at the schedule, give me a minute to look at the schedule. I think we might be a touch behind where we need to be. This is my E&E schedule. Let me pull up the CTI schedule. Yeah, so what we're maybe 20 minutes behind schedule. So chapter 7 is the chapter in your book that deals with genetics. If you guys over the weekend could all take a good look at that so that you guys are well prepared for class on Friday, because I might start picking up the pace just a little bit on Monday. And the other thing is, after we're finished talking about monohybrid and dihybrid crosses, I'm also going to have some practice exercises for you to work to make sure that you know what a monohybrid cross and a dihybrid cross are doing. So I'll also have that ready for you on Monday. We may not have covered everything that you need to know in order to do that, but if we don't finish covering it on Monday, we'll definitely finish covering it on Wednesday. And then the nice thing about Wednesday and Friday is the section on quantitative genetics is actually pretty quick, although there's a little math involved. And then the section of material on Friday probably wouldn't normally take a full hour anyway. So we'll probably catch up and be back on schedule by the end of next week. Regardless of what happens, whatever we've gotten up to through the end of the day on Friday, that's what will be on the exam on Monday. So also think about whether or not you want to have a review session at the end of next week, getting ready for Monday's exam. What I typically do with a review session is I meet with you either on Zoom, or we could do it in person if you want. I can probably find a room that's big enough for us to all be in. And what I do is I come and I'll answer whatever questions you have about the material until I've exhausted all of the questions that you have. And so that's a time for you to get questions answered about the material so that you are sure that you understand it. What that generally requires, though, is that you've spent some time studying the material before we get to that point, simply because if you haven't spent some time preparing, you don't come to a review session with any questions, and so it's just kind of not very productive time. All right, questions?
Are you still going to make the study guide?
Yes, I will put
Yeah.
together a list of the topics that we've covered.
Okay.
But once again,
The process.
it's just going to look like what we've covered in class.
Mhm.
Yeah, Kenton?
The process on the board behind you, is that's the central dogma?
Yes, this is what's commonly
What
known
is
as the
that?
central dogma.
the
Yep.
data transcription to MR image.
Yeah, Ken, I don't know about everybody else, but it sounds like you're
Okay.
talking
That's-
really, really
that's-
slowly. Kenton, are you there? All right, I can't hear you. So, Kenton, stay on for just a minute. Everybody else, I'll see you on Monday.
Hey, Dr. Quincy, you said read chapter 7 for Monday, correct?
Yes, read chapter 7 for Monday.
Okay.
That's the chapter on genetics.
Oh, really? It might be Wi-Fi issues.
Okay. Hey, Kenton, can you hear me?
I'm here.
Yeah.
I don't
Okay,
know
all
if
right.
you can hear me. I have
can.
Wi-Fi issues in Jones sometimes.
Okay, all right. So, why is my what? Oh yeah, here we go. So I'm gonna turn